Hello, and welcome to Chapter 11, which covers sexual reproduction and meiosis. There is an activity associated with this chapter. Take a look on Canvas, and I have a picture here of some germline cells. I also have been playing around with learning how to use the laser pointer, which is fun. I also somehow wrote on this picture and cannot figure out how to remove it. So we're all learning, right? But this is a pretty cool picture. You can see the DNA quite easily, and you can see the different phases these various cells are in. So we have three objectives for today. The first is to understand why animals perform meiosis and what advantage it confers to that species. Right? Second, to know the stages of meiosis. So you should be able to describe what happens in each stage, know the order of these stages. Third, you should be able to compare and contrast meiosis with mitosis. And I think that comparing and contrasting these two types of cell division are the best way to really test whether or not you know what is happening in meiosis. So first, what is meiosis? Why do we have it? Now, meiosis has to do with the sexual life cycle of an organism. And so when we talk about the sexual life cycle, we're talking about two parts, meiosis and fertilization. So meiosis is the generation of gametes, such as egg and sperm, whereas fertilization is the fusion of those two cells to form a zygote. And so that zygote that forms from fertilization grows up. That individual undergoes meiosis in the germline cells to create gametes, and then those gametes fuse when that individual mates with another, and fertilization occurs, right? And then now we have a new zygote, and it just goes round and round, right? Now, a couple of things to make note of during when we're talking about meiosis and fertilization is that somatic cells, non-reproductive cells, have two copies of each chromosome, right? They have homologous chromosomes. And you can see in this picture on the right that this zygote, it's diploid, it has two sets of chromosomes, right? A paternal and a maternal homologue. And each of those individual pairs came from gametes. One came from sperm and one came from egg. And so what that does is that requires that the egg and the sperm be haploid, right? Each one is contributing one chromosome to that pair. And so this allows the zygote, the offspring, to have genetic material from both parents. And this promotes diversity in a population. So why are the gametes haploid? Again, most cells, the somatic cells that make up our bodies, are diploid. They have two sets of chromosomes, right? So we have 23 pairs of chromosomes total, and each pair has two homologous chromosomes. So every time an egg is fertilized by sperm, the egg and the sperm each contribute their one homologous chromosome to the resulting zygote, which is then diploid. If the gametes were diploid to begin with, the resulting zygote would have 4 chromosomes, and that's not the right number, right? And so in order for the offspring to have 2 chromosomes to be diploid, that requires that each gamete, the egg and the sperm, each have only one chromosome and be haploid. So how does this actually happen? How do these sperm cells come to have only one set of chromosomes? And that is what we're going to talk about with meiosis. Right? So meiosis, again, is the generation of these haploid gamete cells. So what happens? Let's see. So let's look at this picture on the right. We have germline cells from parents, mom and dad. Right? The germline cells originally are diploid, and during meiosis, they divide. These germ cells divide, and they produce haploid gametes, egg and sperm. So this clump of cells that we're looking at is a very vague representation of a human body, right? A subset of our cells, our germline cells found in the ovaries or in the testes, are the germline cells. The rest of our bodies are called somatic cells, and they are going to always be diploid. Now these germline cells undergo meiosis, and that is what produces egg and sperm, and these are both haploid. They have one pair, one of each pair of chromosomes. Now during fertilization, when these two individuals mate, we have fusion of those two cells, and that is what results in the formation of a diploid zygote. Now we have both homologous chromosomes. This zygote is going to go through tons of cell division to grow from a single-celled organism or zygote to a fully formed human, right? Again, represented by this clump of cells. And that person, that child, right, as well will have the majority of its body be somatic cells. However, in the egg, in the ovaries and testes, there will be a different type of cell called germline cells. These germline cells are not yet egg or sperm. They have to, what do they have to do? Go all the way back up here and undergo meiosis before they can be haploid gametes. Right? Again, on the right, this is showing in more detail what's happening during meiosis. So one of these germline cells, right, from each parent looking up close, right, the germline cells are diploid. They must undergo meiosis in order to create the gametes that we think of, egg and sperm. The haploid egg and sperm, when they fuse, then result in a diploid zygote, right? And that diploid zygote, single-celled organism, then undergoes many rounds of mitosis to form a full human or mouse or chicken, right? And a subset of those cells in that individual will be germ cells. So again, somatic cells, the majority of the cells in your body undergo mitosis. Your skin cells make more skin cells, right? Your hair cells make more hair cells. But there are certain cells that need to be used for reproduction, right? Egg and sperm. And so to make egg and sperm, those are formed by the process of meiosis. So what we're really talking about today is how are egg and sperm made, right? And so again, a subset of the cells in an individual found in humans in the ovaries or the testes is going to have these properties called germline cells. Germline cells are diploid, and when they undergo meiosis, the end result is haploid gametes. So this egg or sperm, it only has one homologous chromosome. Also, our children are not all clones of ourselves, right, or of each other, right? You don't have all of your siblings and you don't look identical. And so we're going to talk about how there's some genetic mixing that occurs every time fertilization happens, and that allows for a variety of features that you see between yourself and your siblings. So again, meiosis is how haploid gametes, egg and sperm, are created. So how does this actually work? So let's take a step back. I'd like you to think about the stages of mitosis, all right? So in order for a cell to divide, it's hanging out in interphase, right? Specifically in G0. And it gets a signal from its surroundings that tells it to divide. Now, once it gets that signal, does it dive into M phase? No. What does it do first? First, it goes to S phase, where it duplicates its DNA. Right? So S phase occurs before mitosis. That allows the DNA to get duplicated, and then in M phase, the DNA, those two copies can be separated. Now, what would happen if a cell underwent mitosis but didn't first undergo S phase? you would result in having cells that were missing half of their DNA, right? Because in order to split the DNA in two, first you have to duplicate it, right? So mitosis, you start with diploid cells, cells with 2 pairs of chromosomes, and you end with 2 pairs of chromosomes. The resulting daughter cells are also diploid. If you didn't have S phase, what would happen? You'd start out with diploid cells. They'd undergo DNA, the division of the DNA during mitosis. You'd have 2 daughter cells with only half their DNA. You'd have 2 haploid daughter cells. This is exactly what happens, basically, when meiosis happens. So meiosis is very, very similar to mitosis, except that there is a missing S phase. And that is how you're able to result with, start out with diploid cells and end up with haploid gametes. So meiosis contains 2 rounds that basically are very similar to mitosis, but at the beginning of the second round, S phase is skipped. So you think about what would normally happen if a cell underwent 2 rounds of mitosis. If a diploid cell went under 2 rounds, you'd have one cell split into 2, first round. S phase would occur, DNA would get duplicated. Each of those cells would split into 2, and after 2 rounds, you would have 4 daughter cells, 4 cells from that original one. And so the same thing happens in meiosis. You're starting out with one germline cell that's diploid. It splits into 2 during that first round, but then before the second round happens, DNA duplication does not occur. So you have these resulting 4 cells, but those 4 cells each are missing half their DNA, and so you have 4 haploid cells. So before we move on, I want to take a minute to talk about the difference between sister chromatids and homologous chromosomes, because it's really important that you understand the difference between the two in order for you to understand meiosis. So in a diploid cell, somatic cell, we have in humans 23 pairs of chromosomes, or 46 chromosomes in total, right? So each of those individual chromosomes in a pair is called a homologue. Right? In general, each homologue has the same genes as the other in its pair. Now, when a cell gets a signal to divide, it goes through S phase, and this DNA gets duplicated. So this chromosome, homologous chromosome, is copied. We now see 2 pairs here. And this chromosome in the pair gets copied, and we have 2 of them here. Now, that new copy is called a sister chromatid. So we have a sister chromatid here, 2 sisters. Here we have 2 sisters. And these are still considered homologous chromosomes to each other because, again, they all have the same genes on them. Right? Now, let's go into detail into what happens during meiosis. In your book, if you have you can zoom in really close on all the details of this, right? So we're going to start out with a germline cell, a diploid cell on the left. Again, you can see that we have 2 pairs or two homologous chromosomes, right, in here. And during meiosis, we have meiosis 1 and meiosis 2. So meiosis 1 is on this slide, and meiosis 2 is going to be on the second slide. You can see that meiosis has all the same steps as mitosis. We have prophase, metaphase, anaphase, and telophase. Right? But some of the details are different. Right? So we start out with our germline cell, and the first thing that happens is the cell goes through S phase. The chromosomes get duplicated. Right? During prophase, well, you have the typical things. The centrosomes get duplicated, and they start to move to opposite sides of the cell and form the mitotic spindle during metaphase. You can see that the mitotic spindle grabs onto the chromosomes and pulls them apart, right, to opposite sides of the cell during telophase, just like during mitosis. Again, you end up with 2 daughter cells, just like with mitosis. But there are a few key differences. First of all, you see how during S phase we had the DNA duplicated, and so you can see that each of these homologous chromosomes has been copied, and so there's 2 sister chromatids. But the way they line up is very different, right? What's going to happen is that the homologous chromosomes are going to pair. They're going to physically be bound to each other. And so when they line up during metaphase 1, it's going to look a little bit different than during metaphase of mitosis. All right. In metaphase of mitosis, the individual homologs align, whereas in metaphase 1 of meiosis, these paired homologous chromosomes align. Then during anaphase, when things get pulled apart, you can see that these homologous chromosomes are going to be separated, and the sister chromatids remain together. This is different from what we've learned with mitosis, in which the sister chromatids get separated into each of these daughter cells. The last thing I'd like to note that is unique is see how there's some mismatch of colors here? This is called crossing over or recombination. And this is where, again, we said that these homologous chromosomes, they contain all the same genes. And so they are switching over pieces of their DNA, pieces of the chromosome get mixed up and switched around. And so this is able to happen, sorry, forgot to move my phone. This is able to happen because they are paired up like this. And you can see that this variety is maintained all the way down to the end here. You can see that crossing over, you can see that at the end of telophase 1. So we've gone through meiosis 1. The result of meiosis 1 are 2 daughter cells, and they remain diploid. Right? So we're not done because the meiosis is all about making gametes that are haploid. So during meiosis 2, it's quite like mitosis again, right? However, here we're starting with diploid cells and we're ending up with haploid cells. Right? So these 2 daughter cells, the 2 products of meiosis 1, are going to go through basically a second round of mitosis. But S phase is skipped. Right? And because S phase is skipped, the DNA doesn't get duplicated, and you result in half the amount of DNA in each of these daughter cells, and each of these daughter cells is now haploid. Now, this is a pretty complex process to understand, and so I highly encourage you to watch this video that is on the link below that just goes through this. You can kind of stop it and start it, right, and get more details of what happens during crossing over and things like that. And I think especially for this, because it's a dynamic process, things are moving in the cell, having a static image like this sometimes is hard. So check out that animation before you. Here we have a different representation of meiosis as well as a comparison to mitosis. I find that sometimes when I'm studying something complicated, looking at it from different angles helps. So let's look on the right at mitosis because that's what we are the most familiar with. We're starting out with a diploid cell. Right? We've got 2 homologous chromosomes. And first, before mitosis happens, we have the cell going, before M phase happens, the cell goes through S phase, that DNA gets duplicated, and so now each homolog has been copied and we have sister chromatids. These homologs line up during metaphase, and they are split apart during anaphase. 1 sister chromatid goes to each end of the cell, and we result in 2 daughter cells that are both diploid. Again, this is what happens in the majority of the cells in your body in your somatic cells. However, in order to make egg and sperm, meiosis must occur. So we have these diploid germ cells that receive a signal to divide, and they undergo DNA replication. They go through S phase. Each of these homologs gets copied. We can see the sister chromatids here. However, something unique happens where the homologs line up, they pair and attach to each other, and this proximity allows them to undergo crossing over or recombination. Next, these homologous pairs line up on the spindle during metaphase, and then we go through, and these homologous, or these sister chromatids, they stay together. See how they got separated here? They're staying together during this initial cell division. And then the cell undergoes meiosis II, and it skips S phase, right? So we're not copying this DNA, we're just going right ahead and splitting the cell in two again. And now here we can see the sister chromatids finally separate, like what we see in mitosis. What we end up with is 4 cells, and these 4 cells are all haploid. Now, if we did not have crossing over during meiosis, we wouldn't have as much variety in the genetic composition of each of these gametes, right? And so this is why you don't look exactly like all your siblings. Again, meiosis produces cells that are not identical. This is because of that step called crossing over or homologous recombination. And the objective talked about why meiosis is important and what advantage it gives. So we know it's important because it produces gametes, right? But this ability to have crossing over, this recombination, is providing a great advantage, right? Because if you think about natural selection, what's that first rule of natural selection? There has to be variability present in the population. So if I look just like all my siblings, there's no variability. None of us has an advantage over the other in survival. And so nothing's going to change over time. And we won't be able to, our species won't be able to adapt to changing environments. Right? If, for example, you have, I don't know, we have an apocalypse where the earth freezes over. I forget what movie that is, right? The earth freezes over, and one of my siblings has this mutation that allows them to withstand cold temperatures. They are going to carry on the genes of the human race, and we will be able to survive as a population or as a species. But if we were all the same and none of us had any advantage, we would all die and none of us would survive, and the human species would just go kaput, right? And so the ability to have this genetic variety is key to the survival of many species. And this is why sexual reproduction or meiosis is providing an advantage versus species that don't undergo meiosis, so asexual organisms. Right? So meiosis is important because it is what produces egg and sperm. It is also providing an advantage because it gives that species some more genetic diversity within its population, allowing it to adapt to changes in the environment, different selective pressures, and so they have an advantage over asexual organisms. So let's talk about when things go wrong. Right? So non-disjunction is when the chromosomes do not separate properly during meiosis. And so we talked about how the homologous chromosomes get separated during meiosis I, and the sister chromatids get separated during meiosis II. And so we have a cartoon down here that demonstrates non-disjunction, and you can see where these 2 chromosomes didn't get separated, and they both ended up in one of the daughter cells, and the other daughter cell was left without it. So maybe the daughter cell on the left would have 24 chromosomes, for example, and the one on the right would have 22, something like that, right? So if you have extra or missing chromosomes, this is called aneuploidy, right? And this is the most of the time when a zygote undergoes, or when a zygote experiences this, it will not survive, right? This is the cause of most spontaneous abortion. This is occurring super, super early on often. However, there are some situations such as trisomy 21 in which people can live with this and be relatively healthy, right? But in general, if you have pairs of chromosomes not going where they're supposed to, that's not going to be a good sign for that zygote. So let's spend a little more time delving into meiosis and comparing it to mitosis. So these are the four main things that distinguish meiosis and mitosis. First of all, we have homologous pairing and crossing over that doesn't occur in mitosis, right? That's what produces that genetic variety in the egg and the sperm. Second, the sister chromatids remain joined at their centromeres and segregate together during anaphase 1. Third, the kinetochores of sister chromatids attach to the same pole in meiosis 1. That is how they're able to segregate together. And DNA replication is suppressed between meiosis 1 and meiosis 2. So I'm going to go over each of these in more detail. I think this animation, it's a bit shorter than the first one. It's a great summary of the differences between meiosis and mitosis, so I encourage you to check it out. So first of all, homologous pairing and crossing over. This is occurring right away at the beginning of meiosis. So the cell has undergone S phase, whether it's going through meiosis or mitosis. But in meiosis 1, we can see that the homologous chromosomes are pairing up. They're laying right on top of each other. And that's not happening here, right? They're just all kind of randomly floating around in the nucleus. And during metaphase, they line up all separate from each other. But during metaphase 1 in meiosis, they're not lining up separate from each other, they're staying stuck together. They are really close friends, and they're saying, no, I will never leave you, right? Now, what that means is that proximity allows for crossing over or recombination to occur in prophase, or sorry, not in prophase 1, but in meiosis, which does not happen in mitosis. Right? And so during anaphase 1, they are going to segregate together. And so how does that happen? It is because these kinetochores of sister chromatids attach to the same pole. You're like, what the heck does that mean? Let's look at mitosis. We've got our two chromosomes, our sister chromatids, and when the microtubules are out fishing for DNA, they attach to the kinetochore, right? And there's one kinetochore for each sister chromatid, and they get pulled in opposite directions. Now during meiosis, we've got the two pairs hanging out really close together. And so the two sister chromatids together will move to one end of the cell, and the other homologous chromosome containing its two sister chromatids will go to the other side, right? So you can see that basically these kinetochores are attaching to the same pole. These two kinetochores are going and attaching and moving left. These two kinetochores of this pair of sisters are both being attached to microtubules from the right side. And so that's how they're able to coordinate separating these pairs of sisters during meiosis 1 rather than splitting them apart as is seen in mitosis. The fourth point, which I've mentioned throughout this lecture, is that the DNA replication does not occur between meiosis 1 and meiosis 2. So again, normally anytime a cell undergoes mitosis, it will always duplicate its DNA in order to allow it to result in two daughter cells that are diploid, starting out with a diploid cell, ending with two diploid cells. However, if you go through 2 rounds of mitosis, basically, which is what is happening in meiosis, and you skip an S phase, you're going to start out with diploid cells, but you're going to end up with haploid cells because they didn't get a chance to copy the DNA, and so now those resulting cells have only half their original DNA after the cells have divided due to cell division.